Hi, so today we're going to be talking about the genus Amanita. This is some goals and objectives of this lecture. Taxonomic overview, we have approximately 1,000 species in this genus. It is split into subgenus Amanita and subgenus Lepidella, and there are 7 sections divided in between the two subgenuses. One includes the edible Amanitas and one includes the lethal Amanitas. Some adaptations, there's quite a few, but the biggest one is the fact that these are ectomycorrhizal fungal symbionts. So their hyphae network together to form a mycelial layer, which hug the plant's tree roots and actually even penetrate in between the plant's cells. And doing so allows for a symbiotic exchange of nutrients, so it benefits the mushroom and the plant. This picture here shows how it goes in between the cells. The mesh is called the Hartig net, and there's associations that are formed with oaks, birches, beeches, pines, and a lot more other species. Toxic compounds could also be a means of defense or aiding in decomposing material. There is a lot of coordination of growth in relation to water supply and protection against desiccation. Morphology, typically white gills, free gills, white spore print, bilateral lamellar trauma, so the inner tissue of the cap or gill, volar remains, and this can be seen here with the remains as warts or on the pileal surface here. The evolution took place over quite a few eras. So the main point here is that the Amanita genus split off from the Agaricales section in the Jurassic period, and it further split up in the Cretaceous period into Lepidella and Amanita subgenuses, and it further split up, diverged again into other sections, notably lethal and edible Amanitas. The place of origination isn't fully known. However, there is a Gondwana hypothesis, and this cannot be rejected because a lot of the closely related host species have originated in Gondwana, including southern beeches, myrtles, and dipterocarps. Edible Amanitas most likely originated, however, in Africa, in a paleotropical area during the Paleocene and Eocene. The majority of the dispersal took place during the Miocene and Pliocene into other temperate and tropical areas. They had a higher rate of diversification, which occurred in temperate regions of the planet as opposed to tropical regions. This could be due to temperate regions offering more ecological niches in the form of complex soil horizons and a better selection of ectomycorrhizal hosts. The edible section of this genus, subgenus Amanita, shows signs of adaptive radiation, with New World taxa appearing to diverge at a fast rate. So the rate of the Old World was 7.5 million years to diverge, but in the New World, it's not even a million years. So the speciation of the New World was probably triggered by Pleistocene climactic oscillations. The New World taxa experienced lower extinction rates, perhaps due to it being mainly tropical. And the New and Old World somehow have the same species richness, but this came as a result of different events. This is here a picture of an edible Amanita. You can see here the universal veil in different life stages. Section Phyloidae, which is the lethal Amanitas, originated also in a paleotropical zone during the Paleocene. The dispersal, they migrated from the Eurasian continent to North America via the Beringian land bridge. They extended to Central America between the Oligocene and the Miocene. The Beringian bridge travel was perhaps made possible due to climactic deterioration around 33 million years ago, which allowed for more temperate element migration and an elimination of tropical conditions. The section Phyloidae has also been experiencing some adaptive radiation, with rapid speciation occurring in temperate subtropical areas. Diversification may have been triggered by gradual global cooling around 50 million years ago during the Eocene. So this is the current distribution of the entire genus, Amanita. So it's pretty widespread. You can see that it's mostly focused in North America, Mesoamerica, and Europe. And there's also quite a lot of disjunct populations around the world. Limitations and factors of dispersal. So there's geographic barriers like mountains, oceans, deserts. This is within the abiotic factors. Past extinction events due to glacial cycles. This can explain the limited dispersal of the edible section of the Rocky Mountains. Distance plays a role in this. Amanita basidiospores cannot spread over great distances. However, spore migration does explain disjunct distribution patterns between Eurasia and North America displayed in some species. Biotic factors can include limited host availability, so they need to have a host. If they don't have it, they can't really exist. This can explain the dispersal of the edible section in South America, more like lack thereof. Amanita is associated with oaks and pines. Members can only go as far as the Andean Colombian region. People are also playing a role in this. So with a lot of traveling today and industry and exchanges of goods and stuff, the species have been dispersed along with their host species. So an example can be the death cap, which was introduced in North America, New Zealand, Australia, and South America, and Amanita muscaria, which was introduced to Australia. Here's a picture here of the death cap. Looks pretty plain, but it can cause liver and kidney failure. Thanks for listening.